Once upon a time, there was a town called Cell Town. This town was a laid-back and desolate one with little activity. The mitochondria, Mr. Golgi apparatus, Mr. Smooth endoplasmic reticulum, Mr. Rough endoplasmic reticulum, Mr. Nucleolus, Mr. Nucleoplasm, and the chromatin threads lived lonely lives on their own. They hardly talked to each other and kept to themselves, minding their own business. Mr. Nucleolus, however, had bigger dreams for Celltown. He always dreamt that one day Celltown would become a vibrant and prosperous cell city. So one fine day, Mr. Nucleolus decided to make a change, to make a difference. He shared his ideas and dreams with the nucleoplasm, the chromatin threads. They shared similar sentiments with Mr. Nucleolus and decided to work together to turn the boring cell town into an exciting cell city. With their special abilities and talents, they decided to build a control center called the nucleus. Nucleoplasm was in charge of providing space for activity to take place, as well as maintaining the shape of the nucleus. Nucleolus was in charge of storing the secret to producing ribosomes, ribosomal RNA, and also produce ribosomes, which would help to synthesize many important proteins for cell town to function. The chromatin threads, not knowing what to do, decided to lay suspended within Mr. Nucleoplasm. Mr. Nucleolus and his counterparts decided to build a wall called the nuclear envelope around the nucleus using a network of filament proteins called lamina so as to isolate the activities of the nucleus from that of cell town and allow parts of the nucleus to concentrate at the tasks at hand. Tiny gates made out of protein were made in the wall to allow communication between the nucleus and the rest of the community, as these gates would allow the movement of certain substances in and out of the nucleus. At long last, the control center, the nucleus, was finally set up. The nucleus.
The nucleus overall controlled the activities that took place in cell town. It started to delegate tasks to members of cell town. For instance, the nucleus made the mitochondria in charge of synthesizing food substances obtained from outside of cell town to release energy for the community's activities. The nucleus also made Mr. Cell Membrane the guardian of cell town, allowing him to control the movement of substances in and out of cell town. Other than controlling the activities in cell town, the nucleus was also involved in protein synthesis, particularly in the process of transcription.
First, the gene unzips. One of the strands in the gene is used as the template to make mRNA. The mRNA molecule copies the genetic code in a DNA template following the rule of base pairing. The mRNA molecule leaves the nucleus and attaches to a ribosome in the cytoplasm. In the cytoplasm are amino acids and transfer RNA, or tRNA. The tRNA molecules have amino acids attached to one end of their structure. Each tRNA is very specific and attaches only to its own amino acid. Each tRNA also has 3 bases at one end. This is an anticodon that can bind to complementary codons on mRNA. Translation starts with mRNA attaching to a ribosome. The first 2 tRNAs, together with their amino acids, also fit into the ribosome. They attach to the codons on the mRNA according to the rule of base pairing. A peptide bond is formed between the 2 amino acids. Once the peptide bond is formed between the first 2 amino acids, the ribosome moves along one codon to the right of the mRNA. As the ribosome moves to this position, the first tRNA is released. After the first tRNA is released, a third tRNA and its amino acid slots into the ribosome. Then a peptide bond is formed between the second and third amino acid. At the end of the mRNA is a stop codon. This stop codon signals the ribosome to leave the mRNA. Eventually, a whole chain of polypeptide is produced.
Such, the boring cell town soon became a bustling and exciting cell city. Mr. Nucleolus and his counterparts were pleased with themselves and the success of the establishment of Cell City's control center, the nucleus. They even thought of expanding territory and build up more cell cities, but they did not know how. Alas, the chromatin threads discovered that they actually contained highly valuable information that would help in the building of more cell cities.
Firstly, the chromatin threads condense to form chromosomes. Then, the nuclear envelope disintegrates and the contents of the nucleus disperse. The centrioles then move to the opposite ends of the cell, and esters form around the centrioles. Then the chromosomes align along the equatorial plate, and spindle fibers attach to the centromeres of each chromosome. The spindle fibers retract, and the sister chromatids move to opposite ends of the cell. Then the spindle fibers disintegrate and the sister chromatids condense into chromosomes. The nucleus forms at each pole together in the nucleoplasm. Then the nuclear envelope forms around each nucleoplasm. Cytokinesis occurs and furrows form around the two cells. The cells then split apart. Now two cells have been formed.
Many successful and prosperous cell cities were thus established, all thanks to Mr. Nucleolus and his counterparts who founded the control center that started it all, the nucleus.